Welcome to today's Explainer. We usually picture advanced medical tech as this highly synthetic stuff built in massive billion-dollar labs with incredibly complex 3D printers, right? Well, scratch that. Today we're looking at how scientists are literally crossing biological kingdoms to solve some of the absolute biggest bottlenecks in human medicine. And the wildest part, they're doing it using the everyday plants you've probably got sitting in your fridge right now. So how exactly can something as cheap, green, and common as a houseplant offer a brilliant solution to our most complex bioengineering challenges? Let's get right into it.
We're going to kick things off with a question that honestly sounds straight out of a sci-fi movie, but it's actually sparked a total revolution in the scientific community. Can a simple spinach salad help cure human heart failure? I know. When you first hear it, it sounds completely absurd. No way, right? But when we actually zoom in on the sheer mechanics of the natural world, we start to realize that plants and humans have a whole lot more in common than we were ever taught in elementary school. This whole brilliant realization actually happened during a totally normal lunch break. Dr. Glenn Gaudet and his graduate student were just sitting there, eating a salad, completely racking their brains over how to help patients who had suffered massive heart attacks. They looked down at their plates and, bam, an absolute aha moment. A salad. It really just goes to show you that world-changing ideas often come from simply getting curious about the everyday stuff around us, just connecting the dots between seemingly unconnected things. To really grasp why this lunchroom epiphany was such a huge deal, we have to look at the sheer scale of the problem. 7 million. That is the massive number of people who die every single year from heart attacks. When someone has a heart attack, their heart muscle is starved of oxygen, and tragically, it literally dies. Now, modern medicine is incredible. Doctors can go in and place stents to act like the walls of a tunnel, pushing plaque out of the way to get that blood flowing again. But here is the major gap in our current treatment. Fixing the plumbing doesn't bring back the heart muscle that died during the attack. Because of that, the heart just can't pump like it used to, which totally tanks the patient's energy and can eventually lead to total heart failure. So scientists naturally thought, hey, let's just figure out how to grow new human heart muscle cells in a lab and replace the dead tissue. Problem solved, right? Well, unfortunately, no. These new heart cells, just like any normal muscle cells, desperately need a constant supply of oxygen to survive and do their job, which is to beat. To get that oxygen, they need a super complex, microscopic network of branching blood vessels. And here's the incredibly frustrating part. Human 3D printing technology, as insanely advanced as it is today, simply cannot manufacture vessels that are small and complex enough to do the job. But nature? Nature has already perfected this microscopic plumbing over millions of years of evolution. Think about it. The way our bodies transport fluid through our blood vessels is wonderfully, surprisingly similar to how a leaf transports water. To basically borrow nature's microtunnels, scientists use this absolutely fascinating process called decellularization. Now, obviously, you can't just take a raw green leaf and stick it into a human body. Our immune system would freak out and instantly reject it. Instead, researchers take a leaf, insert a tiny needle right into its stem, and pump a simple detergent through it. This detergent acts like a deep cleaner. It breaks up the cell membranes and literally washes away all of those green plant cells. What's left behind is this beautiful, intact vascular network made entirely out of cellulose. And the best part, cellulose is highly biocompatible. We already use it safely in hospitals all the time. This process creates what scientists affectionately call a ghost leaf, and it's brilliant. Imagine taking a regular vibrant green leaf, packed with plant cells, and washing all of that color and cellular material away until you're left with this gorgeous, completely clear, transparent structure. It gives scientists this incredibly complex, safe scaffold that is essentially a clean slate, completely cleared out and ready to be populated with human cells. This isn't just tweaking a leaf, it's completely repurposing a masterpiece created by nature. Now, the scientists had to make sure this wasn't just some fun parlor trick for the lab. They had to rigorously test these ghost leaves to see if the internal plumbing still actually worked. So first, they pumped in some raw dye and watched it beautifully flow through the leaf's entire complex network of veins. Awesome. Next, they leveled up and injected tiny particles the exact size of human red blood cells, and yep, those flowed right on through too. Then came the ultimate test. They injected actual human blood right into the stem. And it worked. The blood successfully flowed down the tiny branching veins of the leaf. This was the exact moment where laboratory theory transitioned into a massive, real-world medical breakthrough. So here's the absolute game changer. Once they knew the plumbing worked, they planted human endothelial cells. Those are the ones that line our blood vessels, right inside the leaf's veins. Then they placed human heart muscle cells on the outside of the scaffold. And because the leaf veins were able to successfully deliver that vital life-saving oxygen, those human heart muscle cells didn't just survive. They attached to the spinach scaffold and they literally started to contract and beat, just like they would inside a human chest. We actually grew living, beating human heart muscle on a piece of spinach. Let that sink in.
This totally changes everything. And once you solve that massive oxygen delivery problem for the heart, a whole new frontier just bursts wide open. I mean, think about it. If spinach can fix a broken heart, what other medical miracles are hiding right now in the produce aisle of your local grocery store? Let's expand our minds a bit and look at how this mind-blowing, cross-kingdom technology is moving far beyond just cardiac tissue.
Researchers all over the globe are now playing matchmaker, pairing the specific physical structures of different plants to specific human tissues. It's wild. They're looking at lucky bamboo as a structure for bone and cartilage repair. They're using parsley stems to create brand new tubular blood vessels. They're taking green onions to model the complex layers of the human uterus. And they're using various other types of leaves to regenerate skin tissue, or even attempting to grow lung tissue in a project that is very appropriately named Lung on a Leaf.
Let's zoom in on that bamboo application for a second because it's super cool. One of the absolute biggest headaches in cartilage tissue engineering is finding a base structure that is porous enough for cells to actually grow inside of it, but also tough enough to bear human weight. Well, it turns out that decellularized lucky bamboo is an absolute structural marvel for this. It has an 86% porosity, and its elasticity level is about 0.9 megapascals. Now, without getting too lost in the weeds, that number almost perfectly matches the elasticity of native human joint cartilage. Basically, bamboo is a ready-made, pre-built house that nature designed perfectly for bone and cartilage cells to move into and start growing.
And the successes just keep rolling in. Moving over to the reproductive system, scientists have been working on 3D modeling of the smooth muscle layer of the uterus. They took these cleared-out plant scaffolds, specifically from green onions this time, and coated them with collagen to help the human cells stick a bit better. And the results? They had smooth muscle cells successfully living, growing, and thriving on this plant-derived structure for over 2.5 months. In the world of cellular engineering, maintaining that kind of viability for that long is a truly staggering achievement. But here's what's truly fascinating about this. The specific architecture of the plant really matters. Native human uterine muscle cells need to align in a very specific, one-way direction to contract properly. When scientists tried growing these cells on a celery scaffold, it was a mess. The cells just grew randomly, totally disorganized. But the outer side of a green onion leaf has these naturally occurring, shallow, elongated channels. When they grew the cells on the green onion, those natural plant channels acted like microscopic train tracks. They physically forced the human uterine cells to line up perfectly in a single direction, flawlessly mimicking native human tissue. Okay, so all of this lab work is absolutely captivating, but as people who really want to understand the real-world impact, we have to ask the critical question: how do these natural, plant-derived scaffolds actually perform in a real hospital setting, especially when you compare them to the synthetic, man-made stuff we've been using? We need to look at the hard clinical data to see why this is such a big deal for actual patient recovery. A recent massive review looked at clinical trials comparing natural, biodegradable scaffolds to synthetic ones in actual human patients. And the standout number from all that data? 0.30. That is the relative risk of tissue recurrence when a surgeon uses a natural scaffold. To translate that into plain English, that is a massive 70% reduction in the risk of a reconstructed tissue failing or having the same problem come back, just by using a natural scaffold instead of a synthetic one. When we break down those clinical outcomes, it paints a beautifully clear picture. Like we just said, the recurrence rate is significantly lower with the natural options. And interestingly, things like how long it takes to heal or how many days the patient stays in the hospital, those showed no significant difference. That means the natural plant scaffolds don't slow down the patient's recovery one bit, but they do vastly improve the chances of long-term success. On top of that, the data showed a highly favorable safety trend for the natural options when looking at complication rates. It just proves that natural scaffolds aren't just some quirky science project. They are vastly superior at healing patients safely and effectively. So what is the ultimate takeaway from all this? It's that humans and plants actually share a deeply profound underlying biology.
By crossing biological kingdoms, we can basically piggyback on the complex structures that nature has already spent millions of years perfecting. From a simple lunchroom realization about a spinach salad repairing a broken heart, to bamboo fixing our knees, to green onions helping us understand reproductive diseases, these inexpensive, superabundant, and eco-friendly plants are literally solving the world's biggest bioengineering bottlenecks. I really hope this explainer has given you a profound new appreciation for the natural world around us. Because to me, it proves that the answers to our biggest, most complex medical challenges might not always require a supercomputer or some multi-million-dollar synthetic lab. Sometimes the cure might just be growing right under our feet. So I'll leave you with this thought. When you start looking at the world a little differently, what other life-saving solutions do you think might be hiding in plain sight, right there in your own garden? Thanks for joining me today, and keep questioning everything around you.